Continuing on with epigenetics, which once again, epigenetics are sequences of DNA, genes that can be turned on, and if they're turned on, they're turned on, and if they're not turned on, they stay not turned on. And this is often dictated by critical moments in lives. I'm going to be taking to this website right here, where it illustrates how this has been shown to affect lab rats. So I'm going to go to this website right here, and hang on. And here we are. This is called Lick Your Rats. Essentially in this simulation, you are a mother rat, and you gave birth to a litter, and you get to now lick your rat pups. What we're going to be looking at is how DNA, which is methylated, which condenses these proteins, can either loosen them so that the genome can be read, or it keeps them tightened so that they cannot be read and transcribed and then translated. So let's click on Go and listen to a little bit of the background scenario.
Deep in the brain of a newborn rat pup, methyl molecules silence the GR gene. When it's active, the GR gene produces a protein that helps the body relax after a stressful event. The type of care a pup receives from its mother during the first week of life can change the expression of this gene.
So, we're going to be looking at the GR gene, and when it's turned on, when it's activated, the GR gene produces a protein that helps the body relax after a stressful event. So we're going to see how we can turn that gene on here with our lab rats. So once I click Start, we'll see the timeline increasing from birth over, seriously, just the first week of life. As I click it, I'll open up a mother mouse right here, and you'll see as I lick and nurture my little rat pup, what that does to the methylation and relaxation of this GR gene right here. So let's check this out. Here's my little mother pup. I'll lick it once, maybe twice. We see that it's loosening up over here. Maybe a third time for good measure.
It looks like you had better things to do, since you didn't lick and groom your pup very much. Its GR gene is still inactive. Your pup will have a hard time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So we notice that this gene didn't loosen up much at all, so it won't be able to be read, meaning that this mouse, as it grows into adulthood, won't be able to deal with stressful situations very well. Let's check this again when mother rat goes a little bit crazy. Oh, look at that methylation. It's really relaxing. Look at that. And uncoiling.
Wow, you're an attentive rat mother. It's surprising your pup has any fur left on its body. Your licking and grooming has seriously activated your pup's GR gene. Your pup will have an easy time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
So as you can see, that is how we can relax these genes, and this was at a critical moment in this little rat pup's life. Within the first 7 days, 8 days, 9 days, 10 days, it doesn't matter. By that point, the damage has been done, and it tends to be permanent. This is actually something I noticed in the hospital when my little lady was born. There's something that they call skin-to-skin contact, where immediately after a child's born, they put it right on the mother, and you have skin-to-skin contact. And I imagine that's to create a bonding, obviously between the mother and child. But also, I wonder if that has anything to do with any of these epigenetics. But it's something to maybe look into. If you're interested in visiting this site, there's a bunch of other interesting information regarding epigenetics, and if you'd like, I have that website on that PowerPoint. We'll go back to that right now. So once again, if you're interested in checking that website out, it's right here. So looking at how transcription initiation, the first step, is started, we can see that on the slide here, chromatin modifying enzymes provide initial control of gene expression by making a region of DNA either more or less able to bind to the transcription machinery. What this is saying is that sections of DNA can be modified to promote transcription from happening. So let's take a look at how we can make transcription a little bit easier to get started. That section of DNA that helps kickstart the process of transcription is associated with what are called control elements here, which help regulate transcription by enhancing the ability of proteins to bind to the promoter, which gets the process started. So we'll have precise regulation of gene expression in all different cell types based on these control elements. Which sections of a gene do we want to regulate? Well, these kind of dictate which sections we transcribe within the genome and which ones we look over, which kind of help us maintain some genetic diversity amongst all of our cells, considering that all of those cells have the exact same set of DNA, but we need to make sure that only certain genes in, say, a heart cell are turned on and other cells in the heart cell are turned off. And that's similar in all cells of our body, dictated by these control elements, which turn on sections of genes. Okay, so this is an image that you do not have in your handout, which we're going to talk about really quickly here before we move on. So before we look at how transcription is regulated, we should take a look at a typical eukaryotic gene, which is what we see on the screen here. Each eukaryotic gene has a promoter, which is right here, and we spoke about the ta-ta box that's at the beginning of the promoter. The promoter is a sequence of DNA where RNA polymerase can bind to start transcription, to start transcribing a pre-mRNA code. Keep in mind that DNA makes mRNA, or I should say pre-mRNA. We have to do some modifications there. RNA polymerase moves in the direction that is said to be downstream, which simply means away from the promoter. So if the promoter is here, we move away from it, we're going downstream. Eukaryotic genes also contain control elements, which we spoke about on the previous slide here, and those control elements are highlighted in gold here, and there's also some up here. These contain DNA nucleotides, these control elements, but these DNA nucleotides, they do not code for anything. So why are they there? Well, their function is to serve as, they provide a place where transcription factors can bind to, which then initiates the process of transcription. These control elements, they are upstream from the promoter, not downstream. They're upstream, so if they're upstream, that means that we're going to be transcribing in the opposite direction. And it also makes sense to say that these control elements are upstream since we want the transcription factors, which are actually controlling transcription, to be on the DNA before the promoter. Notice that some of the control elements are close to the promoter and others are further away. What this means is that not every strand or gene is controlled by something right close to it. Sometimes we can turn on this gene by activating control elements many nucleotides away. I think I read somewhere that sometimes this distance between these control elements and the promoter can be like 50,000 nucleotides. So they can be completely far away from the gene and still be turned on with the appropriate binding of control elements. At any rate, when a control element has triggered the transcription process, initiated the promoter, meaning that the RNA polymerase is able to now bind to the promoter, we make a pre-RNA transcript, also called the primary transcript, with some exons and introns that we need to splice out. And once we splice those out and tack on a poly A tail and a 5' cap, we now have our officially completed mRNA, which is now capable of leaving the nucleus of eukaryotic organisms. So this image here is one that you do have. This is tied to question number 27 in your handout. You might want to pause this for a moment and get some of these words that are highlighted in red labeled on your own sheet. I am going to continue on here. So how do enhancers and activators work exactly? Well, Well, they interact with transcription factors, as we can see in this image. We have our enhancers and activators right here on this control element, and these will interact with these transcription factors, and also a group of mediator proteins, which, as we can see, they all kind of bind to this, which allows the DNA to fold to bring this unit, called the transcription initiation complex, right up nice, snug, and tight to the promoter, which is kind of hard to see right here. And once this is in place, RNA polymerase II can bind to the promoter to start RNA synthesis. Question number 27 is asking you to describe this process that we just discussed, which is also summed up on page 360. If you look at this page, you'll probably find the steps listed in blue helpful, and they're actually numbered in the appropriate sequence. But once again, just to summarize, activators, which are often far away from the promoter, interact with transcription factors and other mediator proteins. These will bind to the enhancer, and with the help of some proteins to allow this DNA to flop over, we can get these units to form the transcription initiation complex, which will bind to the promoter and allow RNA polymerase to bind there as well, starting the transcription of the DNA, making a pre-mRNA strand. So it sounds like gene activation is controlled by maybe more than just one thing. So we're going to look on the next slide at what controls the precise transcription in eukaryotic cells. So here we see two different enhancers, which are these regions right here, which contain various control elements. The enhancers are quite similar. They are found on the DNA strands, and they are quite far from the promoters for the genes that they will transcribe. In the top image, we see the enhancer will turn on the albumin gene, and beneath we can see the enhancer will turn on the crystalline gene. Also, what makes these two enhancers different from one another? Well, to start with, they are made up of different control elements. And I guess the way that this image illustrates that is by using different colors here. Yellow, gray, red versus orange, gray, pink. These different control elements draw in different transcription factors, so different transcription factors and proteins, which in turn will bind to a different mediator protein here to transcribe the DNA. So it attracts different things found within the cell itself. We can see various available activators here, and a particular combination of these activators are necessary for the different genes. The way that this image illustrates the ability of a cell to activate this gene or this gene, it's kind of color-coded to kind of make it easy, but we have red, so we have red here, gray, so we have some gray here, and we have yellow, so we have some yellow here. And when those particular control elements have their activators, those control elements become active, which turn on, essentially, the transcription of this particular gene. Notice that in this one, there is only the gray, but it's lacking the orange and the pink, so we cannot express this crystalline gene. Over here, however, it's the exact opposite. We have the activators necessary for this gene, for this enhancer, so we can activate this crystalline gene and transcribe that one. Notice that the transcription factors and the mediator proteins are quite similar. They're used to draw in RNA polymerase to start the transcription process, but what ultimately dictates whether or not this gene or this gene will be expressed is the availability of the activators to turn on these enhancers.